good afternoon to all of you here in the room and to everybody that is connected with us from anywhere in the world. And I am Gabriella Scarlatti, and I work on the San Raffaele Institute in Milano, and have been working there since many years back. And I'm a partner of EV 2020. And I want that everybody on this panel will introduce themselves. We will have 1.5 hours of discussion, or 1 hour of panel discussion, and I hope that there will be a lot of questions from the audience here and outside. I have one comment. Please, who is not in the room here, use the comment of the Q&A box, and your questions will be then recorded and by I don't remember whom, and will be presented at the end of the panel discussion. So I leave the word to Robin.
Hello, good afternoon, and it's a delight to be here on the panel. My name is Professor Robin Shattuck from Imperial, but I'm the coordinator of the EV 2020 project.
Hello, my name is Hans Langendijk. Is this better? It is better. My name is Hans Langendijk. I work at Janssen Vaccines in Leiden. So my job is I'm head of the vaccine design team, and I'm here as a scientific advisor.
Hello, my name is Hester Kuipers. I'm the executive director for IAVI in Europe. And IAVI's mission is to translate science into public health solutions that are affordable and available for everyone who needs them. And HIV vaccines is one of our key priorities.
Hello, my name is Pepe Alcamí. I'm from Spain. I work at Instituto Carlos III. I lead the AIDS immunopathogenesis unit, working on basic research on HIV. And also, since 3 years ago, I'm the director of the scientific director of the HIV unit at Ospedale Clinic in Barcelona.
Hello, I'm Susan Barnett. I'm from the Bill and Melinda Gates Foundation in Seattle, Washington. There I work on the HIV vaccines and biologics team in the Global Health Division, and I'm happy to be here.
Thank you all. So we have an excellent panel here, and that we have to discuss what's next for an HIV vaccine, so where we are heading. And I would like Robin to start with giving us kind of a sum up, because we had 2 days of fantastic discussions and fantastic presentations of the EV consortium. So I would like him to tell us where we are, what we did, and what's the first things that we should do next.
Well, thank you very much, and we've been having a very exciting and intense meeting here for the last 2 days, looking at what has been achieved over the last 6 years in the EV program, and also looking to the future. And it's been, really very inspiring to look back to when the project first started, and we set ourselves the task of making up to 10 new vaccine constructs. And when we started on that endeavor, many people thought it was crazy, that actually producing just one vaccine from design to getting it into clinical trials would be impressive. And it's been really heartening to see that, in fact, now we have over 10 clinical trials using the vaccines that have been designed and produced by this consortium. Now, we are still challenged to get those trials finished, and it's been an amazing effort to keep the whole scientific endeavor on track during a pandemic, which is certainly something we hadn't factored into our program. Many of you who have been involved in programs often are asked to write a risk mitigation strategy. It wasn't in our risk mitigation strategy, but it's a testament to the dedication of the clinical teams, the researchers, that we were able to continue, as well as many of us contributing to developing COVID vaccines, doing COVID trials. The exciting thing in terms of where we are is the depth of immunology, understanding, and discovery has grown, hugely since the concept of this project. But we are still challenged by the fact that HIV, solving HIV in terms of a preventative vaccine, is a challenge of magnitude that far exceeds solving the development of a COVID vaccine. And so we recognize that there are clear opportunities, but there are no shortcuts to solving this incredibly important but complex problem.
Thank you, Robin. One of the successes of EVI was also the experimental medical trials that no one else uses, really, or have been a bit drastic, but it's a very innovative way of running clinical trials. But it's really, it was possible because only some of the partners could run them, and it's not possible to do them everywhere in the world. So what would you do to convince that this is the way to go, to speed up and become as speedy as COVID has been?
Well, COVID was exceptional because the world faced an enormous pandemic. And huge amounts of funding were thrown at the problem. And that's why things were able to be done in a very condensed time period. But also, for better or worse, commercial companies recognized that if they were successful, there would be a very large marketplace for them. And so therefore, their investment in developing vaccines was de-risked by almost a guaranteed marketplace. For HIV, we know that now the biggest areas of the world that would benefit from an HIV vaccine are in low and middle-income countries. That's not to say there is not a need for an HIV vaccine in developed countries, but the markets are not the same, and the challenges of the timelines are far more exacting. Now, we still recognize that to solve our understanding of how to develop a vaccine that's really effective against HIV will require a much faster rate of progress. And in order to do that, we need to do more clinical trials, whether that's in a conventional framework or whether it's in what we call experimental medicine trials. Now, all clinical trials of a vaccine are experimental because you don't know the outcome. The difference with an experimental medicine trial is not that you are compromising on safety, but you are focusing on a specific scientific question that may help you on your way to achieve a vaccine, but is not focused on developing a product.
Thank you, Robin. I don't want Robin to take the lead and the stage forever, so please.
There's maybe one thing I could add on the risk that the industry took. I remember early 2020 when we thought, well, this is going to be a big problem, but how long is the problem going to take? Because we knew that development timelines of vaccines are awfully long, multi-year programs. And personally, I thought that in the autumn of 2020, everybody would have been infected, and then everybody would have been protected. So it was not an easy decision for industry to step in this, but we just felt a responsibility. And also, indeed, with the help of BARDA, we could pull this off. Nobody ever actually believed that there would be efficacious vaccine so early, and that so many new platforms would be successful. So that was, with hindsight, it seems to be a very interesting commercial opportunity, but it was a big risk, and nobody actually knew how it will develop.
Please step in.
If I see, now I see Susan.
So following along those lines of, how, what was done in COVID, I think what we learned that we can take to HIV, which we should now seize that time, that moment, is that new concepts were tested quickly and broadly And there was iteration, even within programs, and information was shared in real time. And I think that's something all of us should take with us as we move forward. Take some of those concepts that might be on the back burner, might have been tested in the setting of COVID, and start testing them. And to follow on with what Robin said, yeah, get them in experimental medicine trials. Get the good ideas advanced and get the bad ideas killed. Don't just languish in preclinical space with them. So I think this is a really good time for HIV research, and I think we should go more broadly and more diverse concepts ahead.
Yeah. I think it's also good to realize that, here we go, that, and we've seen this with the EV consortia, it is a very strong engine for vaccine innovation, and that's propelling the HIV vaccine field forward, but I think has also had tremendous impact and value in developing other vaccines. So, I think, and then COVID has been able to build on what had already been done in various areas for HIV vaccine research. So I think it's important also for funders to realize that there is a need to continue the development of HIV vaccines because such vaccines are tremendously urgently needed, but also the work that the HIV vaccine is doing and completing and the kind of innovations it needs to develop because it's such a complex challenge is also going to benefit the development of other vaccines, and I think that's an important message for governments and funders.
Yes, I agree that I think it's time for an HIV vaccine. I think that the general opinion is that HIV is over, is not a problem. No? I mean, because we have antiretroviral treatment, so the need for an HIV vaccine is not as important. No? I don't agree with that. No? We have in 2020 1.7 million new infections, 700,000 people dying of AIDS. So HIV is still a big problem of public health in many, many countries. And antiretroviral treatment or PrEP is not enough to block and to solve the problem. So I think that during the COVID pandemics, I would say that for me, the more fantastic thing was the progress, the technological progress. I mean, in HIV, the problem is to design the vaccine. We have not a good design of the vaccine. For the COVID vaccine, I'm sorry, it was an easy vaccine to make the design, no? Because it's an acute infection, a respiratory virus. But I mean, what was fantastic was the investment in new technologies. I'm thinking in RNA and vector-based technologies, and the capacity to generate thousands of millions of doses in a very short time. But the challenge for the HIV vaccine is not technology. I think we are in a previous stage, is to find a good design. What we know now is that if we have this design, the production will be very fast and very efficient.
Can I be provocative? The platforms were there. I mean, if you think RNA was already something that was developed, the vectors were developed, so it was easy to move from other viruses, put the piece of SARS into this, and start producing. If it would have been successful or not, that was another issue. But for HIV, I agree with Pepe. It's not a matter of having the platforms.
Yeah, if you start being provocative, I also like to be provocative because Pepe is right in some respect that it is the design that matters. And I think we came a long way in the HIV field to solve that. And the interesting thing is that for COVID-19, you see vaccines in which the design hardly matters. You have classical vaccines, killed vaccines, even vaccines without too much design, induce neutralizing antibodies and induce effective immune responses, efficacious vaccines. And in the case of HIV, we have wonderful designs that mimic the envelope even better than the spike of SARS-CoV-2 mimics the virus. And it's still not enough. So I remember the field was always troubled by three things. It was the huge variation, the glycan shield, and the instability of the protein. So I think we're going to solve the instability of the protein, but we're just as far off of a solution. You know, we have all these insights, all these technical progress, and we are wiser, but maybe also a bit sadder and wiser, because although the increase in knowledge and design is improved, the huge expected increase in induction of VNA is still not there. So we need to step over one additional obstacle, probably, in order to solve that. Although I think we're on the right track. I don't want to sound too negative. And this consortium will certainly help in this respect.
Robin, your face is...
No, I mean, I think we all recognize the challenges that need to be overcome to realize an HIV vaccine. And I do worry when I see things in the press like Company X is developing a vaccine as if it's going to be done. And that's not the case. Well, there's always serendipity, you may be lucky. But I would say, to counter a little bit, is that although we probably still have a long way to go, we've answered a lot of questions and understand things that don't work. And I think it was Thomas Edison about the light bulb when he was asked. He worked out 1,000 ways how not to make the light bulb before he worked out how to make one. And actually closing off things and saying that doesn't work or this is the challenge is still important. So it doesn't mean that there's been no progress. It just means we still have a long way to go.
And I said there was a lot of progress. Yes. It was a huge amount of progress, but the expected increase in neutralization is still not there. So we need additional technologies, additional insights to master that last obstacle. Yeah.
What does the other part of the room say? Susan, I see you.
Yeah, okay. I just want to highlight another thing, right? I mean, one part is technology and vaccine platform. The other part is measurement. And if we're operating in the dark, when we have a dim light bulb, we're not going to see it. And I think we're in a unique setting right now where we have amazing tools to look at B-cell development over time. We can clone out single cells and study 1 million parameters for a single cell. And so I think we should not give up, because we have evidence that we are on the right track with some of our immunization schemes. And this would be a terrible time to give up, because we have tools so that we can see progress in a way we've never seen before. So on the optimistic side.
Yeah, and following... Yeah, just following your argument, I think that since we started the EV, we have more failures, no, because phase 3 clinical trials have failed, but we have more knowledge and we understand why we have failed. And as you mentioned, the progress, the technical progress and the scientific progress is enormous. But the challenge is very difficult, because I mean, a vaccine, what is doing just is to trigger the immune system to work. No? The problem is, if you have 100 people infected with COVID, 90 will solve the infection. The immune system will be able to cure you. With 100 people infected with HIV, 0 person will be cured by the immune system. So the challenge is to develop a vaccine that should educate the immune system to do something he's not used to do. No, and that's the big challenge, because I mean, the vaccine for other diseases, for acute infections, is able to generate this to trigger this immune response. But for HIV and chronic infections, the viruses have overcome all the mechanisms of the immune system, and we have to teach, to educate the immune system with a vaccine to do a different thing. And that's for me the big challenge. But we had the means. No, I think the progress in B-cell knowledge and in the development of antibodies and is the path to follow.
I saw that you took the mic, but It's not, you didn't want to say anything. You like it. But I was just thinking, in fact, what would we need to do to change? Because it's the challenge is, but how do we do it, right? I know, it's scientific, it's, what do we have to do to change and to get this, what Susan was saying, this, to hit the ceiling and get these neutralizing antibodies?
Yeah, I mean, you know, I think we can do it, right? I mean, we're making baby steps, but we can do it. But, you know, I want to also point out that, you know, we have to think broader. Our field is flip-flopped from antibodies to T cells to antibodies again. And now everybody wants to latch on to RNA. And we tend to have this group think. And I think what we have to do is get, you know, more people involved in the field, not less. And we have to get them, you know, more diverse voices involved in the field. And I really think there should be a strong push to get early career investigators who you know, have a vested interest in using new technologies and exploring new areas. I think we suffer from groupthink, and we suffer from having most of the work done at very few centers. And you know, I think we have to work in Africa, we have to work locally. We can all see how investments in R&D for HIV in African countries really helped with COVID. I mean, with the surveillance and some of the clinical trials that were done in African sites were very important trials. And you know, I think for HIV, we have to even be more mindful of that, to be working in the regions where the vaccines are needed. Because those people, of anybody, are more motivated to come up with winning concepts. So yeah, let's include more people, more voices, more ideas, more regions.
I mean, I would agree with Susan wholeheartedly, but I think we also need to recognize that vaccines could be preventative, but they could also be therapeutic. And the agenda for therapeutic vaccines has also waxed and waned. But now we have the tools to do treatment interruption studies to see how effective vaccines or monoclonals are at reducing rebound, preventing viral escape. Those studies, I think, going forward will be critical to solving the problem, but we need to do more of those studies quicker in order to make the journey to an effective vaccine something that's tangible in a meaningful time frame, rather than a 30-year project from now.
Yeah, I would agree with that, Robin. Thanks for bringing that up. I think, you know, with some of the technologies we're looking at, we could be going into naive individuals at the same time as we go into infected people and do trials to look at not only the immunogenicity of our approaches, but also the potential to suppress virus in an ATI-type study. So I think that's a great avenue for future work. Yeah.
Hester, do you have something to say? Moving into Africa, or we are already in Africa with HIV and COVID, because IAVI has always been working in the African countries, and not only there, right?
Yeah, I think it was a good point that Susan raised. And I think, you know, even if we think about, and we know this from COVID, like how do we think about vaccine equity and ultimately the vaccines that we're developing, reaching the populations where they're most needed. And, you know, that's why it's so essential to be working with communities most impacted by HIV/AIDS and being able to doing a lot of the research there, understanding the sort of local situation, what people's needs are. We know that most people impacted by HIV/AIDS today are not able to benefit from existing prevention technologies sufficiently. So we need to understand what it is that they need and how we address those challenges and really engage those communities in the research. And I think that's going to be essential, not only to propel the HIV vaccine field forward, but also to start preparing for and building for future access and trust among those communities for uptake of those vaccines. So I think engaging there where the vaccine is needed is going to be absolutely critical. I think the other point on, how can we accelerate, I think what COVID was able to benefit from was a tremendous amount of resources, and there was an ability to explore many different avenues. And we were fortunate, as was mentioned, that some of those were successful. We don't have that sort of luxury in the HIV vaccine fields. We sometimes have to take small steps, and then if they're successful, we then have to figure out where the next round of funding comes from. So we're always at a sort of go and stop and go and stop. And we need to also consider how we can generate more impactful funding that is longer term, that is greater in value, and that is sustainable. I think that will be a tremendous way, particularly now where the HIV vaccine field is, to make to have some quick wins and get some accelerated progress. But it requires probably funders to collaborate and come together to put that type of funding up and together and for a durable amount of time. Because if individual funders alone taking those initiatives is going to be difficult. But if funders come together, I think there may be some real interesting opportunities to accelerate this field.
It has always been very difficult to get funders to talk to each other.
No,
but there has been a progress. But
reflect on COVID, right? Billions of dollars were freed up in days, moments, really in relative time. Maybe that's something we have to think about. How do you get governments around this? How do we, there really is not a shortage of money in the world. How do we free up some money to get the resources to do what we have to do? So I think it's up to us to make the case and work with the right people who can influence the people in government to make the money available. I mean, we've been lucky in the US, but we all know we can't count on that forever. We've had a very large budget. In the EU, I think, it's time to wake up all your governments and get them to put up.
EU did something with COVID. They started to work together. It became an EU issue. It was not a governmental issue on single basis, right? So it worked out somehow.
Yeah, but it sure is that it's time for HIV vaccines. But we have to convince, the governments, we have to convince the society also because they will make the pressure on the government. And I think that there are different arguments. One is that HIV is still there and it's a problem. But the second one is that I think we are now developing around the world a third generation of new prototypes of vaccines. That is a really new avenue of research. So we have to convince that it's feasible and it's possible. We have these scaffold proteins targeting the germinal B cells, or some of the prototypes we have developed here in the AV. So for me, excuse me, but it's unbelievable that Europe is not investing in developing HIV vaccines at this moment. No, in this consortium, we have to write every 6 months a lot of papers, a lot of results. Sometimes I ask me who is reading that, no? But I think that no scientists are reading our scientific reports, no? And because if they read these reports, they would be convinced. They will be convinced that it's time for investment in HIV vaccine. I think that Europe is not at the level of investing and fighting for this big challenge that is a global challenge.
I want to know if there is, we are sitting here, but you're here, and you are here to make questions, to say your opinion about all this. And Tom, do we have a mic for Tom? Tom Hankey. Here we have.
Hello. So I want to be challenging as well a little bit, because we've got half an hour of discussion of this distinguished panel, and T cells have been mentioned once. It was all about antibodies and everything else. So let me remind the world that we've got two arms of protective immune responses. Killer T cells are a very important arm, especially against viruses. They kill various factories in the body. They produce cytokines, chemokines. They certainly impose a selective pressure on HIV, and a successful vaccine should make a use of them. And unfortunately, for the last 10 years, development of, or the focus on T-cell vaccine development has been completely missed, apart from few strong-standing individuals. Thank you.
We didn't mention them so that you could stand up. Joking, but go ahead, yes.
Thomas, you make a good point, but absolutely fundamental to this European program has been the concept that you need both. And so it hasn't been ignored in this setting. I take my hat off to you and applaud you for your sticking at it internationally and making sure that it hasn't been forgotten. And I think things change. I think that, you know, I'm a great believer that good results attract good funding. And I think you're at a cornerstone of starting to get the evidence for all your clinical trials. And hopefully that will be game-changing in terms of people thinking about how T-cell vaccines may be essential for long-term prevention and therapy.
Thank you, Tom. Is there any response to Tom here from the panel, besides Robin? You have something? You have a...
I think Robin captured it. And I think Susan also mentioned that the attention goes to T cells, goes back to B cells, goes back to T cells. So I think the field has all the options open.
No more questions from the audience? Oh, yes, there are many. Good. Abby? Go ahead.
Thanks, everyone, for a great discussion. We've got a number of questions that have come through from our online participants, so I'll read some of those out. So I think the first we have is a comment from Mitchell Warren saying, "Thank you, Robin, for calibrating the buzz in the media around recent trials. Important steps, but not yet a product. In addition to building on COVID science, this is a superb moment to build on interest in vaccines and build research and vaccine literacy for the long haul." I guess we could turn that into a question whether the panel would be interested to build upon the recent news of the trials from Moderna in the media.
Go ahead.
I think I prefer to say there is at the moment an extraordinary change in people's understandings about the power of vaccines. And that has been part of the COVID story. And I think we need to leverage that understanding and that excitement to re-engaging people in the search for an HIV vaccine, not to underplay the challenges, not to say that the next trial is going to solve it, because I don't think any of these trials are going to solve it initially, but will be part of the solution. So trials, for example, like the Moderna, are testing very important scientific concepts, which, if they prove to be right, will be a step in the right direction. But it will be misleading to present them as a definitive trial that will come up with an ultimate solution.
You all agree with Robin? Yes, apparently.
I'm not exactly aware of the design of the Moderna. It's the build chief, yeah. So I think the nice thing about Moderna stepping in is that it's going to be really a nice example of getting clinical trial material quickly. So you are the advocate of doing translational science, and RNA vaccines are the platform, the tool to do these kind of trials quickly without too much cost and a reasonable time horizon.
And I was not wanting to criticize Moderna. I think it's fantastic that Moderna are doing a clinical trial. It's fantastic that J&J are still involved in HIV vaccines. We do need the commercial sector to step up and be involved in developing an HIV vaccine, because without commercial sector involvement, if we get a vaccine that works, we will then spend 5 years working out how to manufacture it, because only commercial entities have the ability to provide the solution into the arms of the people that ultimately need it.
I agree very much with Robin. We have to get moving ahead on that part, because otherwise it will again stop. Whatever we get, it will take again ages before it will go into a clinical trial.
Yeah, so don't get me wrong, so normally we don't go into phase 1 if we are not completely sure it's manufacturable. But if we convince them that it's solvable, and then we do like the translational medicine trial for sure, because you can find a solution much earlier before you do big investments.
Nowadays there was a lot of investment of creating new infrastructure for RNA, but not so much for other type of vaccines platform, or?
We didn't have much of an infrastructure for L26, so we built up a huge capacity. We didn't have any capacity when we started. So most of the vaccines came actually from Leiden. So that was a very steep learning phase for us, yeah.
Thank you. I think, no, there is a question.
I just want to follow on the vaccine literacy. That was a very nice term. And it resonates with me because it's right now, people are talking about vaccines. They're saving lives. And it is an opportunity for us to kind of, put an emphasis on that point. If the people don't want a vaccine, we're not going to get funded to make vaccines. So, Mitchell brought that question up. He's an advocate. We have to work with the advocates and keep that vaccine agenda and everybody's in front of everyone. So there are other questions.
Go ahead.
Aus Überla, University Hospital Erlangen. It seems that over the last 5 to 10 years, we've been focusing a lot on systemic immune responses. We focused on systemic immune responses and somehow forgot about the fact that the virus enters via the mucosa. So are there any concepts in this direction to the panel?
We have several people here that worked with mucosa for a while. One is Robin, and but go ahead.
I mean, mucosal vaccination, we know when it's engineered right, works really well. And ultimately, if you have protection at the surfaces that are exposed to the virus and they work, then you're not going to get infected. There are challenges, as you well recognize. One of them is the duration of mucosal protection is often short-lived unless it's a long-term replicating either attenuated virus or some other delivery that sustains the antigenic stimulation. So I think if we ignore it, we're storing up problems. And I think, yes, we should be going back and doing more in that area.
Isn't it also more difficult with the animal models to study real mucosal or in vitro models to start?
It is more difficult. That's the reason why it doesn't get the attention it deserves, for sure. So one of the objections is always that you have difficulty dosing, especially with young kids, with babies. If you try to do nasal immunization with babies, you never know exactly what a dose will be that will come in there. So that's one of the obstacles, and probably also the applicator. It's not going to be a simple syringe. So we think about it continuously, and we always come up with the same objections and problems. So that indeed needs to be solved, because everybody understands that is the port of entry and that is where the defense should be. Yeah.
Yeah. Maybe this is a little provocative, but a lot of old school vaccinologists just say, we protect against mucosal pathogens all the time with our vaccines, and it's just transmutated IgG. And so, you know, that's the counterpoint. You know, do we really need it if we get high enough systemic immunogenicity? So, I mean, I think we should pursue it. Don't get me wrong, but that's kind of what we're up against, I think.
Yeah, but the concentration is so much lower in the mucosa. It's maybe 100-fold lower. So then you need to have a vaccine that's 100 times better.
And we've seen effective regimens for mucosal. You know, you can do the mucosal prime with the parenteral boost or vice versa. I think we should probably pursue that and see if we do better in at least challenge models. Yeah.
You satisfied? Go ahead, there is another question, please.
Okay, we've got some more questions from our online audience. Roger Tattoo says he has his provocative HIV prevention advocate hat on, where he asks, are we doing enough to convince funders and the industry that we need an HIV vaccine, and what evidence do we have that we can put forward?
Silence. Go ahead. I want
That's
comment
my
from
question.
each of you now.
Well, I think we are the only industry that still is in the game, but well, it has been a very frustrating journey, I think, for most of the industry, and because it's so tough, and because there may be agents that are easier to solve, or where people thought the trouble was was bigger, like in the COVID-19 pandemic, and an industry can only do a limited amount of trials, and for J&J, for instance, we have to compete with all kinds of different activities, and we have to spend the money as good as possible. But when we see an opportunity, then we still jump in there. But we feel a responsibility, and that's why, despite the fact that HIV is tough, we really feel that we should try and make an impact. So if there is somewhere out there a good solution that looks good in NHPs or in a phase 1, I think the industry, or maybe with partnerships, they will pick it up, I presume.
Here we go. Okay. Good question, Roger. And I think if you were to look at the European funding picture for HIV vaccine R&D, which has been steadily declining in recent years, I think you could easily argue that we're not doing enough to make it clear to governments and funders why an HIV vaccine is still needed. I think there's a genuine opportunity here, because just like in the scientific community for HIV vaccine research, there is tremendous collaboration. We also have quite an influential civil society network and great collaboration between civil society organizations. And I think we do need to make and continue to make the case of why an HIV vaccine is needed. But we probably also need to look at, you know, how we message it and how we can make the progress that's being made in this field more visible, and potentially having more a united voice of what is needed to achieve an HIV vaccine from a financing and policy perspective, and being able to articulate that well and sort of have, at least on the civil society side, speak with one voice to governments about that. And also maybe a call out to the scientific community. And I know many of you are not only scientists, but also great advocates. And I think, you know, having scientists to also engage more with governments, with decision makers, with funders and civil society, I think is another way to make sure that we communicate well what we have achieved with the money, what we still need, and really making sure that those governments stay committed for the long term.
So I have a quick question, maybe quick or not, we'll see. So I'd like to come back on about the industry now. They dived into the COVID vaccine because basically it was, you know, an easy road, because everything was, you know, never easy road in terms to get public fundings, lots of money. Everyone was panicking, and, you know, it was very supported. And it was, you say, in terms of market, they were sure to kind of get a market. But I think something we forget to mention is, and you can correct me if I'm wrong, but for example, with the EU Commission, it has been basically all the side effects that were possibly, could happen during the vaccine trials or in phase 4 or whatever, wouldn't be the responsibility of the companies, and that the states would have to take that burden and compensate if there were side effects or a sidetrack, something bad happened. So they were also, I think there's this confidence that, they had lots of public funding, then you have the security that you're not going to be, on legal track afterwards by patients. And then you are sure that anyways, the kind of like public health institutions would buy off the vaccine. I mean, the whole thing was just crying out for, just make that vaccine. And this is not what we had for HIV.
I think I already commented that it was not completely clear that it would be a commercial success. I predicted that it would be over in the autumn and the generation of a vaccine would take many, many years. And actually there was not, in the beginning, not a lot of pharma companies who did step in. Pfizer was the only one that did it without any funding. And the vaccine field is very different from all the other pharma companies because you're immunizing healthy people. So even if they get a little itch or a spot on their arm, it is already a problem. So the risk of a pharma company can be huge if you are immunizing 1 billion people and there is a side effect, then it can be the end of the company. So the hesitation, I can imagine, but this was because it was such a global threat. Everybody was willing to cooperate, but I can imagine that the boss of the company does need some help also of others to share possible implication and problems.
I mean, I think that we didn't know the vaccines were going to work early on. And I remember being interviewed by the media probably in January 2020, saying that the chances of success for any one individual vaccine was about 20%. And little did we know that it was actually about 80% chance of success, which was amazing. But it was an acute problem. And I think the challenge that we face in HIV is that vaccine research has been going on for 30 plus years now. And very early in the epidemic, companies did step in because they thought they might be able to solve it quickly. Now, because the route to a solution is not clear, the investment equation isn't there. I personally am absolutely convinced that if anybody came up with a vaccine, let's say, that in a clinical trial showed remission for 5 years, or that gave broadly neutralizing antibody responses against most of the globally circulating strains, you would have no problem in finding a commercial sponsor to pick up that vaccine and develop it for the world. So the challenge is that we know that companies would invest if they were sure that the vaccine would work. But at the moment, the equation isn't there because they don't see that you'll get to the end game in a meaningful time to justify that investment from them, an investment which they have to get a return for their shareholders. That is the reality. And so it has to be a mix of public and private interaction.
On that line, it's working. Even if a company would step in, now, how long time would it take to run a phase 3 with HIV now that we know that we can use PrEP, there is U equals U, so there are many ways. I'm again, putting everybody down to earth. It's not like COVID where everybody got infected and in no time you could really test the vaccine and enroll 30,000 people in a month. And also with HIV we can enroll 30,000 people in a month, but then we have to wait 5 years before we get a readout. So how should we design then a phase 3 to look for efficacy, to be certain and that a company or who else would put the money into it, because it's a long process. It's not like COVID.
I think that's a very good point, and it has not been touched at all, but that was one of the easy things of the COVID pandemic, that there was a lot of circulation, trials were predictable, you could even stop your trial earlier than expected because of the circulation. And I think you touched upon a very, very dark point in the HIV field. Trials will be more and more complicated because you will need to, and of course you want to offer people PrEP, so the trials will become bigger and bigger, and a signal will be more difficult to reach. So I'm not an expert in this field, but I can imagine that it's going to be a major obstacle in the future. So you will probably need a correlate of protection, maybe from another type of study. I don't actually know how to solve
We have
this.
to think about different correlates of protection, or how a different readout, because you have to propose it to a company and say, okay, now let's vaccinate 20,000 people and see, does it work, doesn't it?
I mean, I honestly don't think that it is a challenge. As good as PrEP is, and we know that PrEP and other prevention mechanisms work extremely well, there's still a very high burden of new infections going on in the world. And we've been predicting for the last 10 years that prevention would make it impossible to do an intervention prophylactic vaccine study, and that I don't believe is the case. I think it's still, if we had something that we knew today had a very high chance of working, it could be tested quite rapidly. The challenge more is, when will we get something that will be so persuasive that it will leverage the investment to do the trial? So I don't think the problem is the ability to do a trial, it's getting to a position where it's so clear that this has a really good chance of working that it then merits investment in those studies.
Yeah, and to follow on with that, yeah, I mean, I think there still is a window of opportunity to do an efficacy trial because PrEP hasn't been taken up, but we have to keep in sight that there are long-acting antiretrovirals in development now that could be much more easy to deliver and use, and so that is potentially on the horizon, and it could close the window. I agree with you that we kind of have to captivate a company or the funders or the governments with our ideas, right? We have to come up with something that might captivate people. And it might be in the absence of the ability to do an efficacy trial. And what comes to mind is, I'm just thinking out loud, but, by doing these BNAB trials, these passive protection studies, I'm wondering, to a certain extent, those might provide a kind of a surrogate correlate, orthogonal correlate, so that if we were to induce the B-cell response that we're all working toward, we might have a chance. I'm just trying to offer some hope there.
We would have a cutoff that is set by the AMP trial or by any other BNAB trial that has been working, because AMP has not been working that well yet.
But, Gabriella, I think these are different objectives. I mean, for us, for example, if in a proof of concept trial with 20 volunteers, we develop broad-neutralizing antibodies that neutralize 90% of a large panel of viruses, and of course, strong CTL responses. So I'm sure that a company will be interested in developing that. It's not. I think we have not the capacity to develop a clinical trial to check for efficacy. But if we provide this proof of concept, I'm sure that a company will be interested in going ahead with this prototype.
Do you all agree? They're silent. There, go. Introduce yourself, please.
Joel from Southampton. The stuff about the vaccine companies where they need to be able to make the money back. I was just thinking about with the Oxford AstraZeneca vaccine, how AstraZeneca came in and, to my knowledge, didn't make either much profit or any profit at all. How would you get more companies to do that sort of thing with something like a HIV vaccine? Is that a way, and because that adds another angle to the problem that you're talking about, I think.
I mean, so you have to recognize that the AstraZeneca vaccine is being sold at cost, but it's still the cost of making that vaccine and doing the clinical trials is, more than $1 billion. So it's still required over $1 billion of investment to make a vaccine that could be distributed at cost. And I think that many companies have dual pricing policies. And so it's not impossible to imagine that if we had an effective HIV vaccine, that there would be equitable access. I think it will be easier to provide equitable access to an HIV vaccine than it has been for COVID-19. Because for COVID-19, the rich countries, ordered all the vaccine that was out there. Some of the technologies like RNA were not mature enough to be able to make billions of doses. It's not an acute problem. So I think it's a different problem. But I believe that if we develop a vaccine that can work, it would be able to be made available equitably. I also think that there would be, organizations like Gavi that may well also help to subsidize cost for countries that couldn't afford it.
Yeah, I agree. And, the Janssen vaccine was also for non-profit. It was an example of a private, the private-public enterprise, exactly, yeah. And I think for vaccines that these are going to be very important collaborations, and sometimes the only way to reach a solution, yeah.
I think there is another question from far away.
Thank you. So this is going back to Roger, actually. It's a second part of his question, which says, where is the full public health value of vaccination of an HIV vaccine in a fast-changing prevention landscape? What is the product preferred characteristics that would guide the industry and the blueprint that exists for TB and malaria?
No one wants to answer? Roger. Go ahead. Susie, I'm sure you have something to say.
The full public health proposition for HIV hasn't been made yet. That's what I understand. It's something, I mean, we certainly have put together target product profiles for various HIV vaccines. For example, the one that went, the P5 collaboration, the Alvac prime protein boost, as well as the Janssen Imbokodo regimen, which is adeno prime protein boost. So there were target product profiles. There were initial efforts to put together the full public health value proposition. But I think what happens is when the trials didn't succeed, then kind of the enthusiasm to do that hasn't happened, and they haven't been completed. So if new concepts come up, like, for example, an RNA-based approach or something of the sort, I think we'll have to, as a field, revisit that. But we should be thinking about it, certainly. It's part of the case we make to the policymakers. I really like what you said. I mean, we really do have to, as scientists, start talking to the policy people, the people who can make the economic arguments for us. One thing that comes to mind that we didn't talk about is, the price of the sustaining, drugs, antiretrovirals for all the people who are living with HIV. If the bottom fell out of that, and I worried about that during the Trump administration, have that man in charge of so many lives, politics aside, that could have been a disaster that really showed the world why we do need a vaccine. It's the really only sustainable solution.
There is already an inequality in access to drugs all over the world, so that makes a point to have a vaccine even more, because not all over the world everybody has access for free or low price for drugs, and all the drugs that are available for HIV, by the way.
Yeah, and I think to your point, Susan, we used to have sort of modeling of an impact of a future HIV vaccine that was a generic, it wasn't a particular product, but I think it was a more generic profile. And it is very impactful messaging to policymakers and funders if you can model out the social impact and the social economic impact of an HIV vaccine. And we haven't, to my knowledge, I think haven't revisited that. So I think, you know, it's potentially interesting. The thing, coming back to your point, Robin, about, you know, if we had a proof of concept of a vaccine that would work, we can get industry to come on board and help with what is arguably the most difficult and challenging and financially largest piece of late-stage development. I think that is also an interesting message to funders, where you can say, if you can help us get to a certain point, we are then going to be able to work with industry through the last phase. We do have instruments like Gavi to help with procurement and distribution. So I think in our advocacy messaging, we can probably bring that out stronger, the different pieces in the end-to-end that do exist, and where we would like funders for a particular stage in vaccine development to really focus their efforts. I think it could be helpful. It makes it more sort of tangible and clear where their value would come. So I like that idea.
Roger, do you like the answers? He has time to reply.
We'll wait for his comment on that one. In the meantime, I have a question from Julia McKinday to Prof. Alchemy's comment about needing... Oh, sorry, skipped it. About needing to educate the immune system to do what it is normally unable to do. Is there still a lot that we can learn from the data, from the minority of individuals who are able to control viremia in the absence of treatment, to understand how novel vaccines can be designed to mimic what's seen in these individuals?
Yes, this is an important point. This is a minority of patients that they call elite controllers, long-term non-progressors, and they are a biological model for us, and we study a lot these patients from the genetic, immunological, and biological point of view. You know? And we learn a lot about the kind of response that would be needed to control the virus, because these people are infected, so the immune system is not able to prevent from infection, but is able to control infection. You know? So I think they are a very good model of a study for therapeutic vaccines or for cure strategies. You know? But for prevention, I think would be more the highly exposed and uninfected people, you know, which is a very difficult core to follow. But we study these patients, you know, this minority of less than 100 of patients with long-term control of disease. And we learn a lot of the immune mechanisms that are able to contain and to restrict the viral replication. No? But so, yes, the answer is yes. But again, it's not a preventive mechanisms. They are infected and they control the virus.
But how much do we need a preventive vaccine that would completely preventive without infection, or could we think about having a vaccine that does like the COVID vaccine? People got 3 shots and had 2-3 times also COVID, but they are not anymore sick. So is that something that we can think to develop for HIV, or that's not the case? It's something that, because many vaccines prevent disease and don't prevent infection. So it's not only the COVID vaccine that does this, but how can we do that with HIV? Is that reasonable?
Well, HIV is not a respiratory virus, so I think it's very, very difficult to completely sterilizingly protect the respiratory tract. And so it's pretty difficult to obtain HIV. So therefore, the bar for sterilizing neutralization is also lower. But I expect that you would need sterilizing immunity. And the bar is not as high as for a respiratory infection.
Also because HIV integrates in the human genome, so it would be there and any time could start replicating again if the vaccine is not anymore as efficient. Or
yeah. But anyway, what's needed, we can learn that from the passive immunization studies or other correlates we can get from translational medicine trials.
Robin has something to say.
I think we have to be open to not prejudging how a vaccine would work. You know, if we had a vaccine that could give sterilizing immunity, that would be amazing, but that may be very difficult to realize. A vaccine that reduces the infectious burden and then controls virus so effectively that you don't need to go on to treatment would have a huge impact. A vaccine that prevented transmission from an infected individual, that stopped the virus in its track, could potentially, if that vaccine was broadly utilized across the globe, eliminate the virus within a generation. And I think that actually, while getting a vaccine to work is a challenge, if we can get a vaccine to work, I believe HIV is a virus that could be eliminated from humans over time. And there aren't so many viruses where you could really imagine that a vaccine could lead to elimination in the same way that we hope to see polio eliminated.
Yeah, I agree.
I agree too. You do agree? Down there we have a question. Go.
Kind of following on from
Can you introduce yourself,
I'm
please?
Grace from Southampton. Kind of following on from what you were saying, do you think, if we've got a HIV vaccine that works, do you think uptake would be a problem? Because, you've all mentioned how important it is that I think the West is very naively thinks that HIV is a past problem. And obviously in Africa it's a very big problem, but here in Europe, America, would the vaccine be taken up, especially with all the disinformation that's currently going around? And you just said as well about how it integrates into the genome. Obviously, a vaccine wouldn't do that, but there would be a lot of misinformation going around about that would make people very reluctant to take it up. So do you think that may be a problem that we may face further down the line?
Robin is ready.
So vaccine uptake is always difficult, but I believe that there will be many people who will be motivated to take a vaccine, particularly those people that recognize they're at risk. And we've seen with PrEP that there are some very motivated people that want to take their own destiny into their own hands. But we also recognize that often those people that are most at risk don't recognize that they're at risk. And so the challenge would then be reaching those people that need the vaccine but may not realize that they need the vaccine. But, that would be a great problem to have right now. I would love to be in the phase of we're going after the last few people who we need to get vaccinated. So let's celebrate that when we get there.
Exactly. I think there is more questions over here.
Yes, okay, so during this meeting we've heard about the use of treatment interruption in clinical trials, and so would the panel like to comment on the ethical considerations and the implications for trial participants?
Pepe, go ahead.
Yes, I think that several works show that in people that have a good control of viremia for years, and for example, they are in a trial for HIV cure, no, with a therapeutic vaccine plus TLR agonist and so on. So these trials show that a treatment interruption is not dangerous for these patients because they are set up controls. No, if the viral load increases above, I don't know, 2,000 copies, the treatment is immediately started, and you get control of the virus, and the CD4 counts are recovered. So it's true that in all the approaches of HIV cure, at the end of the day, you have to do a treatment interruption to know if the strategy has worked or not, and it's always a question in the ethical committees. So that's the reason why the people included in these trials are people with good CD4 levels, with a good control of viremia, and that they are under very strict surveillance. No? But the consequences of a transient treatment interruption are not negative for these patients that recover their previous immune status. No?
The controls are always very, very tight, and the patients are followed in a very, very, almost on a daily basis, and that's also a way to overcome any problems. Tom is there with another question.
It's Tom Hanke again. So one dimension we haven't discussed is pediatric vaccinations, because of course success of vaccines is globally in children. So are we missing vaccines by testing vaccines in adults, which don't work in adults but actually might work in children?
You know what I think about it. I'm a pediatrician from the very beginning, and unfortunately we have to go through adult trials before we can go into baby trials. And that's something that I disagree quite a lot. But I don't know from an immunological point of view if we miss something, but it might, because children have a completely different response than not adults, especially in the first year. And that might also play an enormous role, or which vaccine may have an effect in kids. By the way, in kids we can use also monoclonal antibodies in babies. But go ahead.
Yeah, no, there are some trials. I think it's the impact of some trials like this. It's a matter of debate, no? Because, for example, you treat children with antiretroviral treatment, and you get, for example, a lot of parameters suggesting that maybe the control of the virus is very good, no? And then the proposal is, well, we can interrupt treatment in these children. I'm not sure about this strategy from an ethical perspective. For example, it's not a vaccine, but it's also a trial in children. You should be very, very sure that, for example, you have decreased the infection of the level of proviral infection at the very low levels to do interruption of treatment in children. No? So these trials are now ongoing, and I think that the ethical debate is important, because in addition, these trials are being done in Africa. No, are not being done in our countries, because pediatric, HIV infection is very low, fortunately. So I agree with Gabriella that in the case of pediatric research for infectious diseases and for drugs strategies is very difficult, no, from the ethical perspective.
But you can also use monoclonals in case of babies. That's one of the things that can be used and for prevention. And remember that in most of the countries nowadays, mother-to-child transmission is very much reduced at the moment of delivery, but there is still a lot of mother-to-child transmission through breastfeeding, because not every country, every mother can afford to bottle feed their babies. And it's also bound to a stigma, so there we have to intervene. Tom is there, and then Rohir.
How about the adolescent?
Ooh. You're growing in age.
I think, you know, there's another part of this, right? You want to try test vaccines in the populations, you know, that are most at risk because they get the greatest benefit, right? And, you know, and I agree, adolescent women are at great risk, so it makes sense. But there's another aspect to what you're saying is kind of like we don't know the basics yet. I mean, I think we have to learn more about how our vaccines might work in young people versus adults. I mean, there might be a benefit there to early childhood immunization that I don't think we've fully explored, and some people really argue for that. So that could kind of do both things, right? We would understand it, potentially have better vaccines for everybody, plus we could address this issue that adolescent, especially girls, are so much at risk. So I think, you know, I think research in that area should be expanded. And I know certain people are pushing very hard for that. And there are some inroads. And they're testing a lot of this in preclinical models such as macaques.
Macaques.
Yeah.
And baby macaques, juvenile macaques. Go ahead.
This is Rogier from Amsterdam, and I just want to comment on that actually, on NHP vaccinations in infants. And of course, Sally Permar is one of the people who are advocating vaccination in infants. We have vaccinated infant NHPs with a germline targeting SOSIP trimer with fairly good responses. And this month, we're actually starting with a self-amplifying RNA from Robin using the same germline targeting SOSIP trimer. So this is ongoing.
Perfect. That sounds exciting. It's juvenile macaques or newborns.
Newborns.
Fantastic. We are looking forward to the new results coming up from there. Any other questions? Oh yes, here we are. Glad to hear there are so many questions also from outside the room.
Yes, so we have another question. So it's going back to the issue of funding and the discussion we had previously from Mitchell Warren. He says that he agrees with Susan that there is money, and our resource tracking over the last 15 years does show flat funding for vaccines over several years. But with $800 million a year invested each year in HIV vaccine research and development, is it really the lack of more money or how the current 800 million is spent?
I would love to hear from Mitchell what the funding looks like from the European continent versus the US, because I think it's a very different equation.
Yeah. Go ahead, yes, you have that.
I can tell you that. Yes, and you know, and very much sort of acknowledging the tremendous work by AVAC of doing the resource tracking, and it gives us tremendous amount of value, particularly looking over time. And the contribution from Europe to that 900 million has generally been modest. I think around sort of 10% of that at the peak years in 2006 to 2008. And as I mentioned, it has steadily declined, and I think that is a concern. You know, and we do need to, I think, consider, it's been wonderful to have the support of the European Commission for consortia like this, and we've had it for a few years, and also the continued efforts of EDCTP, particularly to engage the Africa continent, Africa researchers, Africa communities in clinical trials for HIV vaccines. So I think that's tremendously valuable, but we want it to continue. And I think with EDCTP and EDCTP3 coming on board now, you know, there are certainly opportunities to get continued support for HIV vaccine trials through European-Africa collaboration. But I think we've also heard here, for example, over the last 2 days, the importance of to continue to invest in the preclinical stages and in innovation and discovery and that early translational research. And I think there is a case to be made towards Horizon Europe now for that kind of money to continue, because we're going to have to bring most promising vaccine trials to EDCTP and into the clinic. And so we need more of those efforts, and I think that's still a case to be made, and Horizon Europe has only just started, and we definitely, I think, need to look for opportunities in its work plan. And there is a commitment in the policy framework, by the way, to support the development of vaccines, including for HIV. So I think we just need to translate that policy commitment into something tangible now. Yeah.
Who wants? Who has, do you have a question? Ah, no, they, sorry.
I'm from Amsterdam, and this is quite a very outside question, but more general for all of us. How do you think the status of researcher and the fact that we're always on short-term contracts, that we have to move all around, that we have to look for fundings, that we're always so insecure about what's next, is impacting the field? Because lots of people at some point get tired of this and they have to leave or change completely, because this is a really exhausting, especially for academia. So I wanted to know how this impacts it, and how the funders, like the BMGF, you can pull out funds whenever you want, and you have like 1-year contracts, although the money is there, and that's very, the uncertainty of it all is not very a peaceful environment to work in. So I just wanted to have your opinions on this, especially for academia, because that's where lots of people, go to private because it's more stable.
I mean, I think we all recognize the challenges of working in academia, but it also comes with, while it has uncertainty, it also comes with a lot of benefits in terms of the freedom to address the questions you want to address, as long as you can justify the science is good science. And I still am a great believer that good science, money follows good science. And actually, Mitchell's question about, do we need 800 million? Is it a shortage of money in the US system? It isn't always that you need more and more money. You need to be smarter about what you're doing. And I think that if you're doing good science, Joanne, you will be supported. But listen, I may look like I'm in my late 50s, but I'm only 23. And that's the toll of going after grants. It's the life we live, and it can't support everybody. But it's also enormously important that we are training people that go into industry, and we often make this judgment that they're two different worlds. They're not. Everybody working in companies want to make medicines that make a difference, but they still need to justify commercially that they do that. Everybody working in academia wants to make a difference, and I've been asking funders for the last 30 years just to give me a blank check. And it hasn't happened.
Never?
So it's like democracy. It's not the best system, but it's the only system we have. And the grant funding system is the only system we have. It's unrealistic to imagine that anybody is going to come along and give long-term contracts. And sometimes that's bad. I mean, we have seen in certain institutions where people don't have to worry about their funding, that they can get lazy. And actually competition for funds sharpens us all, even though it doesn't give you job security.
There is another young one.
I'd like to comment on that. I agree completely with what Robin says, and I think in academia, money follows good researchers, so I wouldn't worry if I were you. And in industry, money follows good products, so the world is not so very different. So you probably spend more time in writing grants, and we spend too much time in making the contracts legally binding, etc. So I've also worked in small biotech, and there the horizon is really small, like a couple of months. So I don't think that you're in an extreme situation. I don't think someone trusts your capabilities, I would say. Yeah, but in the company, the strategy can also very rapidly change. So
yeah,
that's the way it works.
I can add to that. In the U.S., we are seeing an impact. I mean, with all the vaccine science, the successful vaccine science, a lot of people are going, on our grants, we're losing people. They're going to industry. There's certain areas, there's a lot of jobs in industry now. So as far as, funding work, for example, the B-cell work, that's a heavy lift, right? Doing the B-cell sorting and the sequencing. And so, we're kind of thinking, are there contract research organizations that can do that rather than, short-term employees in laboratories, academic labs? So there might be opportunity, for people who, can't get commitments from their academic jobs to go to other types of environments and do similar work. So.
Yes, just a comment, not also to Robin, because I agree that science is a competitive field in academy, in industry, everywhere. No? And I'm also asking projects since 30 years ago to get funding. No? The problem is that competition at the level is positive, but beyond a level is a race of obstacles. No? And then maybe because I work in Spain and Robin works in UK, I agree with what you say, that in fact in Spain we are lose 2 generations of young investigators because we have no the track that they could follow after a postdoc, for example, no? And they have to migrate to other countries or to leave science. No? So I think that there is, we have to balance, no, this competition with our colleagues to be the best, to be very good, but also that we need in our societies that the science to be recognized and specific tracks for young investigators. No? That's one thing that is missing in my country, and this for me is a tragedy at this moment to have lost these 2 generations of young investigators that have no future. No? So competition is good, but an excess of competition sometimes is poisoning.
I think we have last question of today's session. Go ahead.
It was just going to be a comment on all that. It's like we talk about people going from academia to industry and then we kind of lose them. But how do you get people to come back from industry to academia? And then linked to that, why does the academia have to train people to go into industry? Why aren't people being trained in industry and then bringing that knowledge back into academia more? And how can you help that to happen easier, if that makes sense?
We're educators in academia. That is our job. I know we do science, we're doing projects like this, but universities are there to train and educate. So I don't think you can imagine that companies should take on that mantle. There are people that come from companies back into academia. There are people who have gone in and out. It's more tricky because the salaries are usually better in commercial companies. But people are contacting me right now and saying, can you give me somebody who's an expert in RNA? Can you give me somebody who's an expert in formulation? And I have to say, well, actually, they've all gone. Invest as a company in a PhD scheme, and I'll give you an incredibly well-trained person in 3 years' time. And so I would like to encourage more industry investment in training programs so that we can deliver the type of training that's needed. Because we don't want somebody who's done something very esoteric that is actually not attractive in a commercial setting. We need to be training the right people for the right jobs.
So next EV 2020 version 2 will be with a work package 8, that is the training work package, funded by a company, or many companies.
I really like the idea of having postdoc tracks in companies for a limited amount of time, and then circulate back to research institutes, small biotech or whatever, but more of an interaction and less of a divide between the two, I think would be very good for progress of science and for economy and bringing health to people. Yeah.
So I think we have finished. It's 4 o'clock. Our time has gone very fast. I thank everybody, the panelists, very much for the very stimulating discussion and also all the questions, because from the audience and from the audience that we know are outside this room, Roger was very pushy on that. We hope we satisfied the questions. And thank you very much again. And I think this is recorded and can be listened to whenever you want. And it's worthwhile. Listen to it. Bye.